So I want to welcome you to our fifth 4-H QuaranTEEN Virtual Science Café. I'm Laura Wilson. I'm a 4-H science professional. I'm really glad you could join us. 4-H is a community for all kids with programs that suit a variety of backgrounds, interests, budgets, schedules, from in-school to after-school, clubs, camps. 4-H's positive youth development programs are available in your local community. We welcome every kid who wants to have fun, learn, and grow. 4-H is the youth development program of the University of Maine. We're brought to you by the University of Maine Cooperative Extension. So as you take a second to introduce yourself in the chat box, that'd be great. Vanessa's just reminded you in the chat box, we cannot see or hear you. So that's how you communicate with us. So if you share your name and your town in the chat box, here for Maine 4-H, we have Dr. Vanessa Klein and Alice Philbrick who are monitoring that chat. Jessie Brainerd is hosting our Q&A, and she's also working behind the scenes, so all this runs smoothly. And we're gonna keep the session really simple. So our guest is gonna share some of her cool science knowledge. She's gonna tell a bit about herself. We're going to have plenty of time for you to ask and answer some questions. So we'll use the Q&A box at the bottom of your screen to get questions to her. Something different this week, you'll be invited to share some answers to some of her questions in the chat box in addition to a poll question. Speaking of poll questions, I've got a quick one for everybody. And it's just simply, how did you hear about this webinar? So, note about the chat. Would love to see your reactions to what our guest presents and hear your answers to her questions. Try to keep on topic as much as possible. Keep the language clean and appropriate, and we'll have some fun today. So I want to thank you all for logging in so promptly. I'm going to take off sharing my screen. And thanks everybody for voting in the poll and telling us how you got here, how you learned about us. I'm gonna take this opportunity and introduce Dr. Sally Molloy. Dr. Molloy, would you please tell our participants about yourself, the science you work with, and a little bit about your research?
Sure. Hi, thank you for inviting me. I'm super excited to talk to you guys about what I do at UMaine. I'm going to share my screen to get started. Give me a second to do that. All right, can everyone see my screen? Okay. So I am an assistant professor of genomics at the University of Maine, and I'm here in my lab today because this is where I have the best internet connection. I don't have a very good connection at home. And what I want to tell you about is the work that I carry out at UMaine studying viruses that infect bacteria. And I do all this work with students. I have about 12 students working in my laboratory, helping me answer research questions about bacteria and how they impact pathogens. But I also have about 24 students, 25 students that I work with in the classroom that also help me do research on bacteriophage. So I'm going to tell you a little bit about both of those things. And some of the work that I wanted to tell you about today is just things that I thought you would think would be interesting about bacteriophage and how they are master manipulators of bacterial populations. So, oopsie, I haven't figured out how to advance my screens on Zoom. Here we go, this will work. So a little bit about me. This is me, and this is my daughter, Anna, she's 13, and this is my husband, Tom. And all three of us were born and raised in Maine. And I was actually born in Brunswick and I grew up in Bath. So if you're from that area, you know that people who go to school in Bath are shipbuilders. So I was a shipbuilder. And when I was in high school, I knew right away that my love was for math and science. And I actually struggled a little bit trying to decide what area in science I wanted to go into when I went to college. But biology was probably my favorite. So I went up I-95 about an hour where I went to school at Colby College, and I majored in biology, and I loved all kinds of aspects about biology, but I found out within the 4 years that I was there that my favorite courses were about bacteria. So when I graduated from Colby, I went on to the University of Maine, where I received a PhD in microbiology. And I have been at the University of Maine for this fall, I'll have been here for 20 years. So I started as a student. I did a postdoctorate study there with Dr. Ian Bricknell, studying viruses that and bacteria that infect fish. And now I'm a professor in molecular and biomedical sciences, studying viruses that infect bacteria. So that's a little bit about me. Here's some things that I'm hoping that you'll understand from what I talk about today. And some of the things I talk about might be brand new to you. And for some of you, it might be something that you already know a lot about. And so what I'm going to encourage you is to not to sweat the details that you don't understand, and just sort of aim to know maybe a little bit more than you did before this talk. And so some of the things that I'm hoping you'll understand is that bacteria have a huge impact on our world, and they have a huge impact on our health. And so I'll talk a little bit about bacterial populations and how they affect human health. And the thing about bacteria is that they not only have a huge impact on our health, but there are these things called bacteriophage, which we'll talk about in detail. And these are viruses that infect bacteria. And because they have a huge impact on the bacterial populations and what those bacterial populations can do, that means they also have a huge impact on our health. And so I'm going to talk about the different ways that these viruses can be sort of parasitic or predate on bacteria, and that impacts our life. But they can also have these symbiotic or mutualistic relationships with bacteria, and that can also impact our health. And so the last thing that I'm hoping you'll take away from this talk is that students that come to University of Maine to study can be a part of learning about bacteriophage and contribute authentic scientific knowledge to the scientific community. And so these students, on their very first day in college, were able to start carrying out research on bacteriophage. And each of these students isolated a novel phage. Some of those phage end up being used by scientists to do sort of bigger picture things like I do, like looking at the impact of viruses on pathogens. So those are the three goals. A starting place is to understand what bacteria are. And these are my favorite organisms because I think they're very, very talented. So what might be helpful is if they're different than what's called a eukaryote or a eukaryotic cell. So this is a macrophage actually. And eukaryotic cells include like the cells in our body, plant cells, fungal cells, diatoms, single-celled organisms. And there's prokaryotic cells also, and they're a little bit different than eukaryotic cells. And bacteria are part of the category of prokaryotes. So I was wondering if we could start to see maybe if you guys could tell us, I've made a kind of boring poll question here, but it might help start some discussion about the differences between eukaryotic cells and bacteria cells. I don't know if I was supposed to read that out loud. I'm still kind of new at this, but can you guys look at this question? And I've got 4 statements here, and only one of them is true about the differences between a bacterial cell and a eukaryotic cell. Can you guys figure out which one is true and which ones are false?
The good thing is this is not a test.
Oh, good.
So I think we set this up to be anonymous too. So just take your best guess. We've got about 75% of the participants have submitted an answer already. So if we leave it up another 15 seconds, we should be able to share the results with you and show you.
Great. And I would encourage you guys to just take a guess, because in my classroom and in my research lab, my motto is you've got to get it wrong in order to get it right. So can you take a risk for us and have a stab at answering that question?
Yeah, almost 100% have voted. I'm going to
Awesome.
share the results now.
All right. Okay, so let's go through each of these, and then we can use it to each of these answers to better understand things about bacteria. So bacteria are smaller than eukaryotic cells. So that is true most of the time, but it actually isn't true all the time. So bacteria, I'm going to use the word micron, which is a unit of measurement of size, of how big something is. And you don't have to know what a micron is. All you need to know is it's really, really small. But a bacteria might be like 1 to 5 microns in size, depending on what type, how big it is. Whereas like this macrophage here, this is about 20 microns, maybe 25 microns in size. And other cells can be, you know, eukaryotic cells vary, but typically bacterial cells are smaller. But there are examples of bacteria cells that we can actually see with the naked eye. So there's an example called Thiomargarita, and it lives in anaerobic sediments, and it's almost the size of a millimeter, which you can see on a ruler. So that one's not true. It's true sometimes, but but not all the time. Eukaryotes have a nucleus and bacteria do not. So if you use this slide as a helper, you can see that this eukaryotic cell has a nucleus, and this is, of course, is a membraned organelle in the cell where the DNA code is located, right? And bacterial cells, I'm going to address the next statement, do have a genome, and the genome is made up of DNA, and that again is the code. It's like the instructions for how to make the cell and run the cell. They have a genome, but it is not housed in a membrane-bound organelle. So statement number 2 is true, and that of course makes statement number 3 false, that eukaryotes have a DNA genome and bacteria do not. They both have DNA genomes. And only bacteria cause disease in humans. That's also not true. So there's all kinds of diseases, like malaria is caused by Plasmodium, which is a eukaryotic cell. There are diseases caused by fungus. And of course there's, see if I can think of some other examples of, I don't know, there's all kinds of single cell organisms like Giardia that can cause diseases in humans. All right, so let's see where that takes us next. All right, I did define genome. I'm going to use that word quite a bit because what I tend to study are the genomes of bacteria and viruses and how that helps us understand the biology of those cells. So I wanted to make sure I defined that. So my next question is, where are bacteria found? This is a question where you guys can type things into the chat box if you wish. And what kind of environments would you expect to find bacteria? See if I can figure out how to look at the chat box. Chat. Okay, here we go. Give me a second here. I'm going to start reading off some answers as soon as I can see everything. All right. Ooh, I like these answers so far. Mouth, intestines, so different parts of our body. That's true. Moldy food. Yep. They're probably living in there with some fungus. You can find bacteria on your skin. But I see a lot of people answering, human waste is full of bacteria. I really like these answers that say everywhere, mostly everywhere. And so I would actually, let me see if I can make my chat box go away now. I would argue that bacteria are practically ubiquitous, which means it's another way of saying they're practically everywhere. If life can exist on that spot on Earth, then you will find bacteria there. There are certainly places where there aren't bacteria, but that means that there's probably no other life either. So bacteria are pretty talented. They can actually live in places where most other organisms can't, places where it's really hot, places where there's no sunlight. They are metabolically very talented, and it allows them the flexibility to live in places where other eukaryotic organisms cannot live. All right. So why should we care about bacteria? Well, hopefully I'm going to give you some ideas about why we should care about them. One is that they're really important in our environment, and I'm not going to talk about the bacteria in our environment very much, nor the viruses in our environment. They do really cool things, but just as an example of what they do in our environment, they drive really important geochemical cycles. And what I mean by geochemical cycles are probably things that you might have heard about in school by now, which are like the carbon cycle and the nitrogen cycle. Without bacteria, those cycles do not happen. And a good example are bacteria that are the primary producers. These are bacteria that take carbon out of the atmosphere and fix it into organic carbon, which is usable by other organisms. So they're kind of at the bottom of the food chain, and they're pretty important. What I'm going to talk about mostly today, I'm going to move my Zoom boxes here. Sorry. Oops, there we go. What I'm going to talk about today is how bacteria might impact your health and the human body. So most of you guys probably know about bacteria as pathogens, right? Because we talk about things like toxin-producing E. coli and salmonella. But maybe some of you don't realize that the majority of bacteria in our life either live with us, but have no impact on our body, or we actually need them in order to have a healthy body. And so the bacteria that live within our body that make us healthy and make our bodies function properly are called probionts. And so I've got cute little cartoons of these guys because these are the friendly bacteria that live in our bodies. So depending upon which scientists you ask, they will argue that the majority of our body is actually made up of bacteria and viruses. And depending upon which scientist you talk to, they might say that for every cell in our body, there's 10 bacteria that live in our body. Other scientists say there's maybe 1 or 2 bacteria for every cell in your body. So if this represents me, half of these cells are Sally cells, but the other half are bacteria and viruses. So all of these bacteria collectively in our body, that's called our microbiome. And they are kind of like an organism with all the human cells that make us us. And what I wanted to do was to convince you about the importance of those bacteria and how they keep us healthy. So bacteria, we have some questions.
Yep, we had a question coming in the chat. Someone is wondering, does that mean each person is not one organism?
Ooh, I really like that question. And I don't know how to answer it. I think that's all amount of perspective. So maybe you can look at, one of the things that I encourage people to do is to not think about bacteria as a single cell individual, but bacteria are actually populations, and they operate at a population level. And so it's sort of interesting to think that we have many different populations that live within our body. So that's an interesting question. And I don't know 100% how to answer it. It's all a matter of perspective. So you could say that there's lots of different types of organisms that live in our body, but you could also say that they together with our cells make a functional organism. So it's a good question. And I think you can answer it in many different ways and still kind of be right. Does that help a little bit? Deep subject, somebody says. I like it. Okay, so let's see. I'm going to move my chat box a little bit. So the different ways that the bacteria in our body impact our health. One is that all the food we eat, we actually need help breaking down that food. And so microbes in our gut help us do some of that. They have enzymes or proteins that help degrade the food in ways that we can't. But they also, just by taking up residence in our gut or on our skin, prevent pathogens from being able to colonize and take up residence on our bodies. The other things they do is they produce chemicals that pathogens don't like, just by releasing and secreting things that the pathogens say, oh, yuck, I don't want to live here. They help regulate our immune system, and they also make a bunch of metabolites that are like vitamins for us. So these are things that our body doesn't know how to make, but the bacteria are talented and can make those things, and we need them as nutrients. So
we have another quick question for you. I heard a little pause in your voice.
Okay.
Are there more pathogens than probiotics is being asked?
Oh, that's a really good question. And so wow, those I don't quite see come up in my chat box as quickly. So that came up after you asked it. Just so you know, there's a lag for me seeing those questions. So it depends on the state in the human. So if we look at the gut, and again, I'm not like an expert on the gut, but having read a number of papers about it, if there's sort of a dynamic population in the gut, and there can be times, hopefully there's mostly just, you know, probionts that are kind of winning the competition wars, and, you know, edging out bacteria that maybe are not pathogens, but they are something we call pathobionts that don't bother us that much, but under certain circumstances, and if their population gets too big, they can start to cause some harm. And so people who have things like disease like Crohn's disease, or irritable bowel syndrome, tend to have a different population in their gut that is more harmful. Whereas other people who have a healthy gut and don't have diseases like irritable bowel syndrome or Crohn's have a different kind, probably have more probionts living in them happily. Does that help a little bit? And obviously, if you get like an infection like salmonella, or if you were infected with tuberculosis, you'd have different regions in your body where pathogens were able to get a foothold and increase their population. It depends on the particular environment where you're looking at, because different parts of your body have different populations of bacteria. I don't know if that helps a little bit.
And
okay.
We have another question. How does our body determine which bacteria is good and which is bad?
Ooh, that's a really good question. So one thing is, and I don't know, this is where I probably don't know as much about the immune system part, but we are interacting with bacteria like as soon as we're born. Like we get a lot of our bacteria from our mothers. And so our body is sort of developing with those bacteria present, and we're creating tolerance for them. So they are part of helping kind of, our body is sort of learning how to tolerate who's there and who should be there. And then the other part of it, how do we determine if they're bad? So pathogens that we're not used to seeing that come in, our body won't recognize them. And there's different ways, I don't want to get into the nitty gritties of this, but there's different ways our bodies can recognize bacterial or viral components as they are. And they can also recognize them with things like antibodies and T cells. But first they recognize just sort of basic parts of the virus or the bacterium as something that shouldn't be there. That's a really good question. And I really think I can't do justice to it without actually being a super expert on that. And also I probably need like a whole nother lecture to talk about how the body responds to bacteria. But it's a really good question because the body can, for the most part, differentiate the bacteria that are there, that are doing good. And those good bacteria, by the way, if they get in the wrong spot in your body, can cause an infection. So like we have bacteria that live in our body, in our gastrointestinal system. If they got into a cut or a wound, would then become a pathogen and it could do some harm. So it's not just who's there, it's where they have to be in the right place to do good or to do harm. Okay.
All right, we've got one more question.
One more question. Those are really good questions.
And then once some more come in, I'll ask again. This, do we add more bacteria to our system's functions in our lives, or are they all bacteria that have evolved with us as a species to create symbiotic functions?
Oh, that's a really deep question too. These are such amazing questions. I think that, again, I think different people have slightly different microbiomes, but there are a lot of types of bacteria that tend to come up in healthy individuals over and over again. But that's something that scientists are studying right now. And they're really interested in people who don't have a healthy microbiome. Is there a way to shift that microbiome towards one that is similar to somebody who's healthy? But I think there's a lot of work being done on the microbiome right now. And I think there's a lot that we don't understand about how these populations shift. Did I get it?
I'm going to say yes, and I'll let you know when we're ready for some more questions.
Great. And just so you know, like I have the chat box up, but I see them, but it's almost like I see them after you ask them, like they don't appear in my chat box until afterwards. Okay. So one of the things that I wanted to talk about shaping those bacterial populations, one thing that you can do to shape a population in your body is pay attention to what you eat, right? And so the other thing that's important is not only who's in that bacterial population, but what those bacteria are actually doing, and what kind of chemicals they are releasing. So one thing that controls those population is what we put into our bodies. So I have vegetables and here because I think that's what's best for our microbial communities. So if your mom tells you to eat your vegetables, you should totally eat your vegetables because the probiotics really like them. The other reason I'm showing this is because it sort of demonstrates half of the idea that what goes in must come out. And I'm not showing the coming out part, but you guys know that what goes in must come out in organisms. And the same is true for bacteria. So we give them all kinds of nutrients to eat, like whatever's on our surface of our skin, or if they're in our gut, what we eat. They're taking in those nutrients, they're metabolizing them, and they are releasing chemicals too. Okay, and so those chemicals that those bacteria release have a huge impact on our body. And so some scientists have discovered that some of those chemicals that they release look like the neurotransmitters that are in our brain, that might signal happiness or sadness. And so those get into our bloodstream and actually can go into our brain and impact our mood. Those chemicals can stimulate and regulate our immune system. And again, they also add all kinds of metabolites and nutrients that we need in order to live. So I think that the point that I really wanted you to understand is that there's microbes that are in our body that have a really important function in keeping us healthy. And I'm still having trouble advancing slides here. So who is in that microbiome is important to our health, right? And the other thing that's important is what kind of chemicals that they're making. So what they're doing and who's there. So the question I think I want to give you a chance to reflect a little bit is what kind of questions do you have about bacteria and how they affect our health? And you can ask questions about the bacteria that make us healthy, but I tend to know more about the bacteria that don't make us healthy. And I'm going to talk a little bit about pathogens in a little bit too. But if this is a place where you guys want to ask some questions, you can ask questions here too. And if not, I'll move on.
We did have the question come into the Q&A. Is that why it is said that chocolate gives you the feeling of being in love?
Ooh, I don't know the answer to that question. All I know is chocolate is delicious. I don't know how that translates to feeling in love, but it's possible that it's impacting some bacteria in your gut, and that the bacteria are making some sort of chemicals that go into your brain. But I have no idea what the connection between chocolate and feeling in love is. But it's an interesting question. Any other questions before I move on? Because I'm going to talk about the ways that viruses impact bacterial populations.
We've got, what is your opinion on the usage of hand sanitizers because they eliminate healthy bacteria?
Oh, you know what? There is a woman at Colby College, and she studies the microbiome of the skin. And I can't answer for her, but she is an expert on all the different microbes that make our skin healthy. And she says a lot of the products, I don't know specifically about hand sanitizers, but certainly if you're using hand sanitizers, that is going to have an impact on the bacteria that are on our skin that actually protect us. But she would probably argue that any of the products that we put on our skin, from sunscreen to makeup and all those things, could potentially nudge our skin microbiome in a way that's not particularly healthy and make us more susceptible to things like eczema and rashes. I'm not an expert on the skin microbiome, but certainly anything that we put on our body will impact those populations. But in this current climate right now, when we have to weigh introducing into our microbiome viruses and bacteria that can make us really, really sick, then you kind of have to weigh the risk of you know, touching something that has a flu virus or a coronavirus on it that could possibly make us sick. And then maybe using those sanitizers is probably a pretty good idea.
All right, we've got 2 more questions, and then after those, whatever comes in after that, we'll move on to the next question session. We have the question, if bacteria affect your mood, are bad bacteria related to mental illness?
You know, that's also a really good question, and I probably can't answer that, but I would probably say that people who study the gut microbiome might say that if maybe that what we eat can help improve maybe how we feel, but I'm sure that there's a lot more to it than that. That's a little bit out of my area of expertise. It's a lot of it out of my area of expertise.
Okay, and our last question for now, can bad bacteria hurt good bacteria?
Yes, that's a really good question. I wasn't going to go into that very much in this talk, but some of my research is looking into bacteria interactions and how actually viruses that live in bacteria can help certain bad bacteria have what's called an antagonistic, or kind of like a mean, harmful interaction with another bacterium. So yes, but likewise, sometimes the good bacteria can have a harmful interaction with a bad bacterium. So bacteria have all kinds of tricks to edge out their competitors. That's another really good question. Can I move on a little bit?
Yep, I say move on, and then we'll more questions when you're ready for another question break.
Great. Okay, so now we're going to get into an area where I definitely feel more I will probably feel a lot more comfortable answering questions, and that is the question of what controls who is in a bacterial population and what can those bacteria do. So this is a question using a chat box. Can you guess what has the biggest effect on a bacterial survival in any environment? And I'll give you a hint. It's my favorite thing to talk about. Other bacteria can certainly do that. That's a good answer. We just actually talked about that, right? That bacteria can be antagonistic to each other. And that's absolutely true. Food can impact who's in a population. Temperature, right? So different environmental conditions are going to allow some bacteria to be more successful than others. What else do we have? Bacteriophage. Okay, so that is the surrounding environment. So all those things are really good answers, and all those things do play a role. Weather, waste, but probably the biggest threat to a bacterium is infection by a bacteriophage. And so we're going to talk about how bacteriophage shape bacterial populations. A bacteriophage is a virus that infects bacteria. And I'm going to argue that they can be like a predator, or I'm going to use also the word parasite by killing and lysing bacteria. And they can also be like a symbiont, where they live within the bacteria and they actually help the bacteria survive. So we'll define those terms a little bit more. One of the things that I want to make sure that you understand is, before we get into that, is what is a virus? And viruses can be really, really simple. They can be a genome, which is made up of DNA or RNA. So here's a virus that's just like the helical DNA genome and made up of nucleic acid. And the only other component to this virus are proteins that are kind of protecting that genome. And so this is a very simple virus. But there can be other viruses where here's the genome and it's some sort of nucleic acid, either DNA or RNA. It's covered with a protein coat. But some viruses also have what's called an envelope, and this is made up of a lipid membrane. And sometimes they have these little spiky things, these little spiky proteins coming out of them. So my question for you in the chat box is, can you give an example of a virus that you know something about? And this should be a pretty easy for you guys to come up with answers to this. And you can answer about viruses that infect bacteria or viruses that infect eukaryotic cells. Flu virus, COVID, try to think anything but COVID-19, somebody says. COVID-19, a cold virus, so like a rhinovirus. Cancer is not a virus, but there are viruses that can trigger cancer. They can cause cancer. Those are called oncoviruses. And there's different types of oncoviruses. The Spanish flu is influenza, flu. Yeah, you guys are coming up with the scary ones. All right, so you guys know lots of examples of viruses that infect eukaryotic cells. And so because we're in a pandemic, I chose a picture of the coronavirus. This is COVID-19. And this is a virus that infects cells that are in bats, but also cells that we have, right? So within our respiratory tract. This is actually a diagram showing SARS, which is related to COVID-19. And it comes inside the cell and it releases its genome. It uncoats and it replicates its genome. And then it packages up all of its new genomes and makes a whole bunch of new particles, and those are released from the cell. And usually the cell from which they're released doesn't fare very well. It usually dies, right? So that's, those are viruses that infect eukaryotic cells. And of course, there are also viruses that infect bacteria, and those are called bacteriophage. I'm seeing somebody added mad cow disease. Mad cow disease is actually caused by a prion, which is actually a protein, a rogue protein. You guys have all kinds of great ideas about viruses. All right, so a bacteriophage has a capsid protein. So here's a capsid head. The genome is inside of this. You guys can see my mouse when I'm swinging that around, I hope. The genome is inside of this head, and then they have these cool tails and this lunar landing gear, which helps it find its bacterial cell hosts. They come in all kinds of shapes and sizes. So here's some electron micrographs of different types of bacteriophage. And they're a little bit different than eukaryotic viruses in that they stay on the outside of the cell and they inject their genome inside the cell. And then once inside the cell, they replicate this genome and they make a whole bunch of new progeny or baby phages. And most of the time, but not always, they are released by lysing the cell. And so here's a picture of a bacterium that's actually been infected by a whole bunch of phage on the outside of its cell. And on the inside of the cell, you can see these dark spots are new progeny phage that are forming, and they are released by pretty much blowing up the cell to smithereens and releasing all these phage, and they go off and infect new hosts. I'm gonna check the chat box again here. All right.
Are you ready for, we do have a couple other questions from.
Let's have them.
Okay. We have going back to bacteria with the questions about food. We have someone wondering, what type of food would we have to eat to feel happier?
Oh, vegetables, anything with lots of fiber. And that's what the gut microbiome people say. Lots of high fiber diet.
Okay.
Lots of veggies.
And then we have someone is wondering, why in summer does bad bacteria go away?
Sometimes they do and sometimes they don't. It depends. So viruses, sometimes bad viruses go away that don't fare well in like hot weather. So some viruses, bacteria tend to do better in the summer, but viruses, particularly the ones, remember I told you some of them have those envelopes, those lipid envelopes like the influenza virus. They don't like to be really hot and they don't like to get dried out. So the virus doesn't stay what we call viable, meaning it can't cause an infection if it's exposed to those really hot and dry conditions. So if I sneeze and I get virus all over this environment, if it's really hot and dry, maybe the virus will kind of degrade more quickly in the hot weather. So that's one reason why and the other reason is people have windows open and they're outside more. So that's one reason why they think that viral diseases kind of slow down in the summer. It's a good question.
Okay, one question. What does it mean to LYSE? I'm not sure the pronunciation, so I'm going to leave that.
Lice. Oh, it's a good question. I'm going to go back. This is a cell that has not lice. Can you guys see my mouse when I use it?
Yes, we can. Yeah.
Okay, so this is a cell that has not liced, but it's gonna lice. This is a cell that's liced. It's blown up. It's no longer a cell. It bursts open. That is what the word, that's a great question. That's a word that somebody says explode. Yes, Vanessa says. Okay.
All right, I'm going to ask you one more question, and then we'll hold the rest till the next question time.
Okay.
We have the question, what is the worst virus in history?
Ooh, that's a very good question. The worst virus, probably the pox virus, smallpox. That is a very deadly disease, and we don't really have to worry about it anymore because through vaccination, because of this virus only infects humans, and they were able to develop a really good vaccine for it. We no longer have smallpox pandemics, but that's a very, very deadly virus and made a lot of people very sick, and most of the time it killed you. So somebody says Black Death. Black Death was actually caused by Yersinia pestis, and that is a bacterium. So that had a pretty big impact on populations too. Right now, my vote is for coronavirus. But there's all kinds of, there's a lot of viruses out there that have caused a lot of death. Like if you get it, then most of the time you will die, but they aren't very transmissible, meaning they don't spread from person to person very easily. But there's lots of nasty viruses out there. But there are also lots of viruses that you have in your body right now and you don't even know it, and because they don't make you sick. Okay. Anything else, Jessica?
We have a few, but we'll wait till the next stopping point so you can do this.
So there's a couple things I want to help you understand about bacteriophage. One is I want you to better understand how they shape bacterial populations, and how they might shape your microbiome, but also how they impact pathogens that try to make us sick, because that's what I study. But I also kind of want to tell you a few things about bacteriophage that I hope will make you think that they're super cool. So that's where I'm going to take a little detour and talk about why I think you should study bacteriophage. I think that there's 10 to the 31 reasons why you should study bacteriophage. And the 10 to the 31 comes from the number of bacteriophage particles that scientists estimate are on Earth at any given point. And 10 to the 31 is kind of hard to like wrap your brain around how big that is. But what it means is that there are more bacteriophage on Earth than any other biological entity. So organisms, the bacteria are the most abundant organisms on Earth. And they estimate that for every bacterium, there are 10 viruses. And there's a scientist whose name is Chris Settle, and he studies viruses in the ocean. And what he did was take a picture of one droplet of seawater, okay? And he stained it with a stain that only sticks to double-stranded DNA. So any organism or virus that's in that droplet of seawater that has a double-stranded DNA genome will fluoresce under a microscope, okay? So what they did was they counted up all the big things. So like this big thing right here is probably a diatom, and my chat box is covering my screen, but maybe this big blob over here is some sort of single cell organism. But all these medium-sized points of light, those are bacteria, and the pinpricks are bacteriophage. So let me tell you a little bit more about that number to see if you can, I can tell from the chat box, you guys are trying to wrap your brain around how big that number is. So here's some back of the envelope math that scientists have done to try to understand how big that number is. So if they know how many bacteria there are and they think that there's 10 to 30 on Earth, if there's 10 bacteriophage for every bacterium, then that's 1 order of magnitude greater, 10 to the 31. And I really like this. Somebody says, OMG. That is such an appropriate response to how many bacteriophage are on Earth. Okay, so Chris Settle decided to try to make that a little bit, translate that to us so we can try to understand how much mass of viruses there are in the world. And again, he likes to talk about viruses in the ocean, but there are predicted to be even more viruses that, for example, live in the soil. But here's some back of the envelope math that Chris Settle did. So he figured if each virus particle is about 100 nanometers in length, he calculated how much mass there was just in carbon in a particle. And he estimated it was about 0.2 femtograms, which is so tiny. It's like 10 to the minus 15th of a gram. So that's kind of hard to imagine. But if you add up all the predicted particles, virus particles in the ocean, that adds up to be 200 metric tons. Okay, well, that's a big number, but I don't really know what a metric ton is. So he translated that into whales. So blue whales are the biggest mammal on Earth. And if you added up all the viruses just in the ocean alone, that would be the equivalent of the amount of carbon in 75 million blue whales. So I don't know if you know how many blue whales on Earth there are, but there are not 75 million. There's more like 10 to 20,000. So that is a lot of mass. Another way to think about it, if you stack these viruses from head to toe, from tail to head, that would end up being like 10 trips back and forth across our galaxy. That's how many viruses there are in the ocean alone. Okay. So how do they impact our health? That's the original question that we had. So I kind of took a detour there. Remember, my argument was that they're going to be kind of like parasites to bacteria, but they're also going to impact bacterial populations by being mutualistic or symbiotic. In order for you guys to understand that, I've got to ask you another chat question. In order to understand how phage can do that, you have to understand what is the major goal of any life form or biological entity. Viruses aren't really alive, so we'll call them biological entities. What is the major goal, do you think, of every single life form on Earth? And I'll give you a hint, including rabbits. Can you guys put in the chat box what you think the goal of every single life form or virus is? Okay, you guys have nailed it. Survive and reproduce, survive and reproduce. You've got it. And everybody's typing that in. Awesome. Okay, so if you keep in mind that viruses are just like everybody else on Earth, they just want to make more of themselves. Okay, so turtles do it, bacteria do it, and viruses do it. Their goal, if you want to be immortal on life, you have to make more of yourself. Otherwise, you disappear off the face of the Earth. And so those that are most successful of making more of themselves are going to continue to be on Earth, right? So here are how phage do that. How do they make more of themselves? They do this in 2 different ways. They do it in a way that would come under the category of parasitism or predation, depending upon, which I'm not going to differentiate these 2 for you. But the bottom line is they do it in a way where they benefit, but the bacterial cell that they infect does not benefit, right? So in comes their DNA into the genome. They replicate their genome. They make more of themselves, and they make more of themselves at the expense of the bacteria. The bacteria dies, right? So this is a form of either predation or parasitism, where the relationship between the 2 is that one really benefits, the virus is benefiting because it's making more of itself, but the bacterium no longer has the freedom to do that, so it is not benefiting. All right, so how does that impact bacteria that might be on our skin, that might be in our gut, living on our body? Well, let's just take a simple system here where we have 2 different flavored bacteria, the red kind and the blue kind, okay? And the thing about viruses is that they can infect some bacteria, but not all bacteria. So a virus that, for example, can infect these blue bacteria, if this is one species or strain, probably can't infect the red kind. So what happens to the blue guys? Well, most of them die, probably not all of them, but most of them die. And when they die, because the bacteria infected them and lysed them, that gives the red ones an edge and an opportunity because they're not being infected by bacteria. They can reproduce and use all the nutrients the blue guys didn't get to use because they died. So if you think about if the blue guys were a pathogen or a pathobiont, so basically a bacteria that would cause us harm, then maybe these phage made it our guts a little bit healthier. Or maybe the phage were infecting a bacterium that when it died made room for the red guys if they're a pathogen or a pathobiont, and this could cause disease in the gut. So that's one way that viruses can impact bacterial populations on our bodies, is simply by choosing who wins in a bacteria and gets to make more, a population and gets to make more of themselves. I think I have a pause here. This would be a good place for a pause.
Okay.
The next.
Are you ready for some questions?
Yeah.
Okay. The first one I've got here is, do bacteria cause allergies?
Do bacteria cause allergies? I don't know 100%, but I'm going to give you my best guess. I'm going to guess that they don't. And the reason why is because usually our allergies respond to, like our IgE, our antibodies that cause allergies, usually respond to things in the environment, especially like for parasites. So parasites can ramp up our IgE. I don't know how they could cause allergies. I feel like I can't answer that question really well. It's a good question.
Okay. How about, what is the difference between good lipids and bad lipids?
Good lipids and bad lipids? I don't know. Maybe like, are we talking about the type of lipids that we eat and how it impacts our health? That's actually way out of my area of expertise. If that's what you're asking, what kind of lipids should we eat? Like what kind of fats? I only know what my doctor tells me.
All right, here's one I'm pretty sure you'll know. How many, or you will be able to answer, how many different types of viruses are there?
Oh my word. So it turns out that this population, which I told you is really vast, right? They're the most abundant thing on Earth. They're also incredibly diverse. So because they can replicate so quickly, their genomes change quickly. I don't have that in this talk because I thought it would be beyond what you guys would want to know, but they're so diverse. It's actually what we study with our students is the incredible diversity. They are big gene swappers. They swap genes with bacteria, they swap genes with each other. And so we call their genomes very mosaic because it's almost like they're made up of bits and pieces from all kinds of different things. So they're so diverse, it's incredible. Great question.
Okay, and the last one I've got for now is, what do bacteriophages do when they come in contact with skin cells?
Probably, I don't know, but I'm guessing not very much. So like a bacteriophage cannot infect a skin cell. It can only infect certain bacteria, and it can't even infect all bacteria. It can only infect bacteria that have certain things on their surface that they recognize that will allow them to get their genome in. So bacteriophage can't really hurt us, but they could hurt the bacteria that live within us, if that makes sense. Are we good?
Yep, we're on to the questions.
Okay, great. So how can bacteriophage impact bacteria like a symbiont? And this is close. Now we're getting closer to what I like to study. So I told you about how bacteria can be parasitic or predate on bacteria and kill them, right? But they have many bacteria can have a different type of relationship with bacteria, and they can cause a different type of infection. So this is the lytic infection that leads to lysis. But there's also something called a lysogenic infection, where the bacterial genome, which we're seeing here in blue, it actually gets integrated into the I'm sorry, the bacteriophage genome is in blue. It integrates into the bacterial genome. And so now we have one circular genome that includes a virus genome and the bacterial genome. So what happens now? Well, the integrated genome is called a prophage, and the cell that harbors the prophage is now called a lysogen. And the majority of the bacteria that live in your body are lysogens. They have these little prophage living inside them. And now we know that viruses like to replicate themselves, right? That's their biggest goal in life, is to make more of themselves. But they do it when the bacterial replicates, it replicates its genome, and it also replicates the phage genome. And so now there's a copy of the phage genome in each of these two daughter cells. And so you can think about it as the success of the prophage, or the virus, is now tied to the success of the bacterium. So it shouldn't surprise you that through this evolving relationship between this virus and this bacterium, the prophage has learned to bring a few tricks that help make the bacterium survive better in the environment. I did want to add that these bacteria that carry these integrated prophage, they can undergo something called induction and go back into the lytic. I'm not going to talk about that much today. I'm going to talk about the fact that these viruses that live inside the bacteria will express genes, meaning they're making proteins that the virus codes for in its DNA, but the bacterium does not make those proteins. And a lot of times those proteins it makes will benefit the cell. I'm going to move my chat box a little bit here. Fit by protecting the cell from more phage infections, because remember what the biggest threat to a bacterial cell is being infected by a phage. Well, if there's a resident phage there, it will actually protect the cell from infection by other viruses. And it also will express other genes that might give the bacterium superpowers. And so it might encode a toxin that makes the bacterium a better pathogen. It might encode an enzyme that helps it use a nutrient in the environment more efficiently, or it might make the cell more resistant to antibiotics that are also found in the environment. So this is a symbiotic relationship. And what it results is that with cells that have this prophage, so this little pink represents the prophage here, and the prophage is helping the cell make these little pink dots, which are special proteins. And cells that make those special proteins are going to be more competitive than cells that don't have the prophage and don't have those proteins. Let me just do this real quick, and then we can take another question break. So this sort of summarizes it. What controls who's in the bacterial population and what those bacteria can do. Now we have a situation where our symbiont, which is the prophage and the bacterium, both are benefiting. The phage is benefiting because it has a safe place to live inside the bacterium. And the bacterium is benefiting because it's going to be more successful in its environment compared to bacteria that don't have that prophage. So everybody wins. Now, if you think about it this way, this bacterium that has the prophage is going to be able to do better in the environment and build up its population compared to this other guy. Now, the next thing we have to think about is this guy carrying the prophage, is it a pathogen, or is it one of the cells that lives in our gut that protects us from pathogens? And I actually am going to switch over to talking about how prophage make bacteria better pathogens, because that's what I study. But I'm going to take a break here to see, I don't know if my next slide is I'm going to take a break here to see if you have any questions about that.
Sure. I have a question from ISIS has heard about biophages being used for genetic engineering or the delivery of a CRISPR system. How do they do this if they cannot infect eukaryotic cells? Apologies for the pronunciation. Okay.
So, so we use viruses. We do use viruses, particularly in bacteria so we like to engineer bacteria, meaning like we like to add and subtract genes in the bacteria. It helps us understand what those genes do and learn more about the biology of the bacterium. There are some bacteria, so E. coli is very easy to work with. We can put DNA in E. coli very easily and basically manipulate the genome of the bacterium very easily. Other bacteria, it's very hard for us to get DNA inside of the bacteria. It's something called competence, how well that bacterium can take up DNA from the environment. And so with those types of bacteria, and Mycobacterium tuberculosis used to be one of these, where the way that we can manipulate the genome of mycobacteria is by using viruses that can infect the cell, get that DNA inside the cell, and basically it acts like a molecular tool for us to manipulate that mycobacterial cell. So that's one way that we use viruses as tools. Somebody asked about CRISPR. CRISPR is actually a bacterial system used to protect the bacterium from viral infection, from bacteriophage infection. So we use the genes. I actually use CRISPR in the lab to manipulate bacterial gene expression. And we use enzymes from bacteria. And I don't want to get into the nitty-gritty of CRISPR. It's something that's used to protect against bacteria, but we use it to manipulate, to make very specific changes in the bacterial genome, or those same systems can be used because they're just the genes for the CRISPR system. We can put those genes into like a zebrafish cell or a whole organism and manipulate in a single cell the genome of that organism. So we're not actually putting the virus inside that organism, if that makes sense, but we're using the genes from bacteria to make those manipulations. Does that make sense?
Dr. Molloy, I want to do a quick time check because we're a little bit after 4 too.
Oh my gosh, it's already 4 o'clock?
They've had they've had such great questions today.
I'm gonna skip this stuff. And I'm just gonna tell you real quick that I study viruses that infect bacteria. And this is a cool story. I study viruses that infect bacteria, and what I'm looking at is how they make those bacteria more antibiotic resistant. And so I study a relative of Mycobacterium abscessus. This is a bacterium that makes people very sick, especially if you have cystic fibrosis. So I can't tell you too much about this, but we can use viruses. I don't do this, but this scientist right here, his name is Graham Hatfull, and he uses viruses to treat people who have infections to Mycobacterium abscessus. And these infections are completely resistant to antibiotic resistance. Are completely resistant to antibiotics. And so this scientist named Graham Hatfull has figured out a way to treat this little girl. Her name's Helen Spencer, who they thought she was going to die of this infection, was able to treat her with phage and reduce the infection in her. And so now she's happy going to school again. The other thing I was going to tell you, this is my one research slide where I study how the viruses make bacteria more antibiotic resistant. That's what my research is about. And the final thing I want to tell you is that all the viruses I use in my lab and the same viruses that are used in Graham Hatfull's lab, who is the leader of mycobacteriophage research in the world, is that these viruses that we're using were isolated by students like you in college. And so if you come to the University of Maine, on your first day of school, you will come with a soil sample and we will help you isolate viruses from the soil. And you will isolate it, characterize it. We'll even sequence the genome and learn about the genome of that virus. And you get to contribute your work to a real authentic scientific database that real scientists are using to do real science. So that's my plug. Here are a bunch of my students from my classroom and also from my lab where they've isolated phage. Here's some students doing electron microscopy. And here are students presenting at real scientific meetings down at Mount Desert Island Biological Labs, but also at the Howard Hughes Medical Institute here. They're giving an oral presentation of their work. Here are those same students, and they're here with this actually very famous phage biologist, Dr. Graham Hatfull. And so if there's any more questions, I'll take them. Sorry, I ran over.
Yeah, I've got one more question in here. Is a virus in a person like a virus in a computer?
Perhaps in the sense that it's it's a parasitic relationship, but obviously a virus in a computer, which I don't know very much about viruses in computer, but a virus in a computer is not a biological entity, so no. I'm gonna answer it that way. But the relationship's the same.
And that's it for questions.
Great.
And just to make a note, there is somebody in the chat who says they have an aunt in Utah who studies bacteriophages and is studying the possibility of phages that attack cancer cells.
Very cool.
This has been fantastic. I really appreciate you coming out, Dr. Molloy. It's been really a pleasure to have you explain all this to us. I think the questions today were amazing. Been really.
I'm so pleased with the questions. Some of them are a little I was a little shaky on. I was a little happier when we got to the phage part, but these were really obviously very thoughtful, well-informed students. So
they're fantastic. I'm going to ask those of you who are still on the line, I know a couple people had to leave, but most of our participants are still there, to fill out the quick evaluation poll that I've put up. As well, if you have other topics that you are interested in having us bring a guest in to talk about, drop a note about those in the chat box. We've got a couple more topics coming up over the next couple weeks that we know we have scheduled. We have Dr. Jason Bolton next week. Dr. Bolton is a food safety specialist, but he also teaches this cool thing called innovation engineering, which is a way to take an idea and make it into something big. So if you've ever thought, hey, this would be a really good product, or somebody should make something like this, or I could make this idea into a business, you should tune in next week. Registration's available on our website right now. I'm also going to quickly share that website with you guys to learn more about 4-H in Maine. You can simply come to our 4-H website. The other cool opportunities going on right now at that 4-H Learn at Home website, which also has all of our science cafes on it, is that we now have a bingo contest going on. So if you go to that Learn at Home website, you'll be able to access a bingo card, and there are prizes. And you do not have to be a 4-H member, and you do not have to be in the state of Maine to participate. So I'm pretty excited that my colleagues have put together a bingo session. And just as a reminder, we've got an upcoming event online Tuesday, May 5th called Don't Fall Into the Thinking Trap. And this is by teens, and it is for teens, and it's about keeping your mind from getting stuck in one of those traps. So for more information, you need to register by Friday, but I've got Sarah Conan's email up there, or you can call her and she can give you the information. That information is also on our website. I really appreciate you all coming out today. This has been a fantastic afternoon, and really appreciate Dr. Molloy. Thanks for taking your time out of your day to do this.
Thanks for having me.
All right, and remember, if you have other types of science cafes that you'd like us to offer, drop a note about what kind of science or engineering you're interested in the chat box, and we'll see if we can make it happen. And everybody, have a great week. I hope to see you here next week, and wash your hands often. Bye, everybody.